Hello and welcome everyone to this podcast. In this podcast, we're going to continue our discussion of reprogramming epigenetic modifications. Remember, we've already talked about somatic cell nuclear transfers, and we've talked about cell-to-cell fusions. We want to talk about two more in this podcast. The first one is reprogramming by the use of cellular extracts, and then a technology called induced pluripotent stem cells. But we're going to begin with cellular extracts. Alright, so reprogramming by cellular extracts. So this can be done in a variety of ways. So let's start by taking an epithelial cell. What happens first is that the cell is permeabilized. And it's done by using a toxin that we're going to just call SLO. It stands for streptolysin O toxin, but we'll just call it SLO. And then what happens here is it punches a bunch of holes in the membrane, and now we can apply cellular extracts to this cell and see what happens. So the first thing that was added, or rather I should say one of the early experiments, I don't know if it was the first experiment, but it was certainly one of the earlier ones, they added T cell extracts. So in a separate cell, a T cell, they lysed it and isolated the cellular extracts. They then bathed this permeabilized epithelial cell with those extracts. And they ended up with a cell that now expressed T cell genes, indicating that this original cell could be reprogrammed to behave sort of like a T cell. Now, it wasn't functionally a T cell. It didn't automatically have T cell properties, but it began to express genes specific to T cells. So this early experiment was really important because it established the concept that we could reprogram the cell. They also took these permeabilized cells and added to it the extracts from embryonal carcinoma. So these carcinoma cells, these cancer cells from an embryo, the extracts are bathed in this epithelial cell, and it generated this cell. And these cells, after 1 week, they expressed OCT4. And remember, that was that important transcription factor that turned on a lot of genes necessary to maintain that pluripotent state. So this is now expressed. And of course, this gene is not expressed in the epithelial cell. B-type lamins replace A-type lamins. And B-type lamins are the ones specific to pluripotent stem cells. The gene for OCT4 is demethylated. For a better sequence, I should have mentioned that first. And then lastly, epithelial genes are off now. So the specific transcription factors found in this stem cell, essentially this carcinoma cell, is able to convert this epithelial cell to a cell that resembles the embryonal carcinoma, and functionally it behaves as a pluripotent stem cell. So the third experiment I want to talk about is they took mouse embryonic stem cell extract and bathed the epithelial cell in it. OCT4 becomes demethylated, and we have this biphasic expression of OCT4. So down here we have time, and over here we have OCT4 expression. And within 1 hour, it begins to rise. And then by 24 hours, it peaks. And then by 36 hours, the levels drop to almost non-detectable levels. And then it lags here for a little bit, but then it starts to rise again. And by the time you get to 72 hours, it again peaks. It will remain at this high level here for weeks. These cells will behave as embryonic stem cells. All three of these examples explain that there's something in the cell extract, even if you take a differentiated cell, a T cell, or if you take an undifferentiated cell, the embryonic stem cell, if you take those extracts, they are able to change the genome of this donor cell to match the cell where the extract came from. There are likely transcription factors, perhaps various histone modifying enzymes or histones in them that can replace what we see in this case in the epithelial cell. Now let's take another look at this, but from a slightly different perspective. This time let's start with an embryonic stem cell, and we can permeabilize it to create this cell with holes in it. Okay, we can now take this permeabilized embryonic stem cell and add to it cell extract from a differentiated cell. For instance, we could add cell extract from a pneumocyte. These pneumocytes, just so you know, they are cells in the alveoli, which is a part of the respiratory system found in the lungs, and these cells are responsible for gas exchange. Clinically speaking, these cells, being able to generate these cells, would potentially have some use, either in research or perhaps for some therapeutic purpose. So the idea is maybe you could add cell extract from the pneumocytes to these embryonic stem cells, which, remember, are undifferentiated, so maybe we could reprogram them. And so what happens when we add this to it, we push differentiation towards a specialized cell. In this case, we're talking about pneumocytes. So we'll push them towards becoming a pneumocyte. Now, this is anything but efficient. As you can imagine, as you add the extract, you're adding everything that's in the cytoplasm of the pneumocyte. And so it's not a very accurate way to measure precise amounts. But we do see some differentiation. So in addition to trying to find a way to make a source of these cells, it also allows us to study this pathway here, going from undifferentiated to differentiation. And it allows us to determine which factors from the pneumocyte are necessary to create the specialized cell, the pneumocyte in this case. Knowing those factors may at some point allow us to, with more specificity, cause this embryonic stem cell to differentiate into cells of a specific pathway. Okay, now let's move on to this last topic, or last example of how we can reprogram a cell. So these are called induced pluripotent stem cells. And these for short we call iPS cells. Now remember, up to this point we've talked about cell-to-cell fusions and the use of cellular extracts. Remember, with the cell-to-cell fusion we had limited success. We were able to get reprogramming of the donor cell, but that tetraploid state caused the problem. Even if you were able to maintain the tetraploid state, the cells that were produced from that would be hard to use because, again, they're a tetraploid. Another problem, if you remember, is those tetraploids specifically lose the somatic DNA. It's reprogrammed initially, but then it's lost because it's lacking some of those cis factors that are likely very important in establishing the pluripotent stem cells. Now, using the cellular extracts that we just talked about, these are a little better because we avoid problems associated with creating that tetraploid. However, it's still a very inefficient and limited process. And they don't differentiate to the extent that would be useful in a clinical setting. But this new technique that we're going to spend the rest of the time talking about, called induced pluripotent stem cells. So let's talk about those more specific. So I'm going to describe for you a few different ways that we have created these iPSCs. The first was back in 2006. This was the first documented evidence of producing these cells. And essentially what we're going to do is we're going to take a somatic cell and make a pluripotent cell out of it. But in this first experiment, they used mouse embryonic fibroblast cells. They then added to these fibroblast cells a retrovirus, which are very useful tools to deliver specific genes to cells. And this retrovirus contained the following genes that could be expressed in the new cell. OCT4, which is the transcription factor that we've talked about previously, SOX2, KLF4, and c-MYC. And what resulted were cells that resembled mouse embryonic stem cells, those pluripotent cells. They looked like them, so they had an appearance, or we could say morphology, that was similar to the embryonic stem cells. Growth rate, similar to embryonic stem cells. Gene expression, similar to embryonic stem cells, and different than the original fibroblasts. And it gained epigenetic marks on the genome that were consistent with an embryonic stem cell or embryonic pluripotent cell. They did a microarray experiment. Microarray is just a good way to compare gene expression in different cells. So you could compare the genes being expressed in this iPS cell with a typical pluripotent cell and see if there are any similarities. And what they found were several similarities in gene expressions that made them almost indistinguishable from an embryonic stem cell. I should also say that they did these experiments using mouse adult fibroblast cells to show that even a differentiated cell could be reprogrammed. A more differentiated cell could be reprogrammed. However, when they took these cells, kind of in a classic experiment, they would take a blastocyst stage shown here. Remember, it has that inner cell mass, which will go on to form the embryo. And what they would do here is they would add iPSCs to the inner cell mass. If these iPSC cells can behave as a pluripotent stem cell, then they should incorporate into ICM. And then when you produce the progeny, it should be a chimera. It should contain genes from both the blastocyst stage of this donor cell plus genes from the iPS cells. And when they did this, it would have been great if I could have told you a different result, but you got to go with the data you get. And their data was that no chimeras form, suggesting that these induced pluripotent stem cells did not become fully integrated into the inner cell mass and ultimately differentiate into endo, exo, and mesoderm. Even though this part of the experiment failed, it was an important first step showing that we could reprogram a differentiated cell into a cell that was essentially an embryonic stem cell. But there was still quite a bit of work that still needed to be done. So this original group, as well as some other groups, went back to the drawing board to see if they could generate a more stable iPS cell. So essentially what they did next was they would take this somatic cell, they would add to it the same cells as before in the retrovirus, so still a retrovirus. So it contained OCT4, SOX2, KLF4, and c-MYC. They hypothesized that one of the reasons this retrovirus wasn't working as well is that the retrovirus had a hard time entering the cell. So what they did was they took these somatic cells before they gave it the retrovirus and they introduced into it a retrovirus receptor, one that could specifically bind to the retrovirus to allow for a more efficient incorporation into the somatic cell. This turns out to have helped quite a bit, and this enabled for a more efficient reprogramming. We also saw, using this method, the ability to form ectoderm, endoderm, and mesoderm. So having this receptor allowed this retrovirus more efficient access to the somatic cell. It allowed for a better reprogramming of the genome and as a result, this cell would divide and differentiate. They also discovered if they added to this retrovirus the transcription factor that we've talked about before, nano G. And if we did this, we were able to generate chimeras, I should say in mice, where you're able to take this blastocyst, which contains the inner cell mass, and add to it these cells here and allow it to develop. And they end up producing an individual that contains genomes from this inner mass cell and genomes from this somatic cell. In addition to using retroviruses, they've also used a virus called lentivirus. This has using a lentivirus, they were able to show that just OCT4, SOX2, and nano G were all you needed to convert this somatic cell into an iPS cell, which could then be used in a blastocyst to generate these chimeras. In addition to these 3 genes, sometimes they would add a 4th gene called Lin28, but it's not essential for this to occur, but it does increase the efficiency. So really, all that we need to express in these somatic cells are these 3 genes, OCT4, SOX2, and nano G. Those 3 genes alone will reprogram the somatic cell nucleus. The next thing they want to do was to show that iPS cells could be made from a variety of donor cells. Partially specialized cells, fully specialized cells. They wouldn't be able to show that we could make iPSs from almost any cell type. Now we don't know if we can make them from any cell type, but we do know if we take the following cells, they can be converted into iPS cells. Okay, so some of these cells include fibroblast cells, which is what we've talked about on the previous board, pancreas beta cells, liver cells, and stomach cells. And there are other examples too, but I'll just stop here with these examples. Now, all of these can form iPS cells. When you make these iPS cells and you introduce them into blastocysts, they will form these chimeras. And in particular, we can see the somatic cell DNA and the DNA from the blastocyst. We see those in the germ layers that will go on to form gametes. Another fairly interesting observation is that iPS cells taken from liver and stomach cells are less tumorigenic, much less likely to form a tumor. So let's talk a little bit about the actual reprogramming. We don't know a lot about what's happening, but what we do know, I think we should talk about. So remember, we're going to take our somatic cell. We're going to introduce to it using a virus, either the lentivirus or the retrovirus. But the take home is we're going to add to it OCT4, SOX2, nano G, Lin28, and KLF4. Now we know we can take only the first 3 here and we're able to get it, the induced pluripotent stem cells to form. But let's keep these all down listed here because I'm going to define something about them in just a moment. We know that these iPS cells that are form at low frequency. Very few of all the cells that get this retrovirus, only a few of them actually become an iPS cell. Most of them die. This suggests that these genes that we're introducing into the somatic cell, so the trans genes introduced, don't reprogram the genome universally. We know some of the genes that are demethylated for sure, so not this OCT4 and not this nano G, but the OCT4 and nano G that are found in the somatic cells. We know that these are demethylated. We know that this demethylation of this iPS cell is much slower. Remember, if you take an oocyte and fertilize it with a sperm, the sperm DNA that enters this cell is immediately demethylated in a very efficient and fairly rapid response. But the demethylation in the iPS cells is much, much slower. suggesting it has a harder time to demethylate that DNA. So how are the chromosomes in iPS cells demethylated? Because remember, we want that. If they're going to become embryonic stem cells, the genome needs to be largely demethylated. So let's remind ourselves what happens during normal fertilization. During normal fertilization, once the diploid nucleus is formed and it needs to be demethylated, one way that's accomplished is keeping DNMT1 and 3 from the nucleus. They're trapped in the cytoplasm. So these enzymes that add methyl groups are kept out of the nucleus. Thereby they are not able to just randomly methylate targets. We know this can't happen in the iPS cells. So let's see, this is fertilization. In the iPS cells, this can't happen, and let me explain why. So here's our nucleus. Our nucleus was over here as well. We know that we need DNMT1 and 3. We know we need those to enter the cell's nucleus. We know that's important because somatic genes, those genes found in this somatic cell that allow this to be a differentiated somatic cell, those somatic genes have to be turned off, downregulated. One way they can accomplish this is by having DNA methyltransferases enter the nucleus. And we know in iPS cells, DNMT1 and 3 are in the nucleus. So how does it manage to demethylate everything else? The answer is we don't know. But we do know that it is an extremely slow process. So it's possible that it takes a long time to demethylate the DNA when you actively have DNMT1 and 3 in the nucleus. Whatever the reason, we know it's a very slow process compared to fertilization, which is a very rapid process. So we don't really know how it's happening, but let's take a moment to describe some of these genes that are found here and what they might be doing. So c-Myc is important in cell proliferation, cell division. We also know that c-Myc helps to acetylate histones. This acetylating of the histones may help turn on pluripotent genes, those genes necessary to maintain the pluripotent state. Next, let's talk about KLF4. Oh, before I move on, c-Myc, I should also remind you, this is an oncogene, so it's not surprising that we see cell proliferation, but it is also a cautionary for us to think about the fact that c-Myc may be the reason that sometimes we see these iPS cells forming cancer cells. Alright, so KLF4. This protein, when expressed, decreases p53. We don't want to completely eliminate p53 because it's a very important tumor suppressor gene, but it's important that it's downregulated in this case, because when it's downregulated, nano G is upregulated. KLF also blocks pluripotent differentiation. The last one I'm going to talk about is called Lin28. Lin28 is an RNA processing protein, and in this processing of various targets, it helps maintain pluripotent state. Okay, that's all I have for this podcast and induced pluripotent stem cells. If you have any questions at all, please make sure you come talk to me. If not, I'll see you in class. Bye.